Good morning. How are you doing today? I'm doing well. I'm doing well. Today we're going to talk about the electrophysiology of the heart. You ready?
Sure.
Okay. All right. Well, first thing I'm going to do is draw a brief anatomical view of the heart, not to scale, of course, and then I will talk about the different kinds of tissues that you have on the heart. Okay, so basically you have two kinds of tissues. You got specialized and contractile tissues. Okay, specialized tissues do what? Special functions. You're very correct. What does the heart do that other tissues generally don't do? Exactly. The heart generates its own electrical impulses, and it's automatic, right? Even if you take the heart out of the body, it still continues to beat as long as you provide it with the right nutrients and oxygen and all that. So what generates the automaticity in the heart? Yes, correct. The SA node and the AV node. There's also two more. Sodium potassium pumps, yeah, they generate the membrane potential. We'll get into that, but the two other things that I'm talking about are the bundle branches and the bundle of His and the Purkinje fibers. Okay. Now, out of these, the SA node is the most important one. It's called the cyanoatrial node, and I'll get into it. Okay. So let's see where this is situated in the heart. All right. So here you have the heart. Okay. I'm just going to draw it like this. All right. And I'll divide it up into 4 chambers. Now, of course, you all know what these chambers are. What are the chambers, by the way? What are these two? The atrium. Very good. Atria. And these are ventricles, exactly. Now, where exactly is the SA node? Yes, exactly. It's somewhere up here in the right atrium. And then you have your AV node. Where would you guess that would be? Yeah, it's somewhere around here. And then you have your bundle branches, or the bundle of His, which goes in through your interventricular septum, the wall between the two ventricles. Okay, and then it sort of curves around like so. And so this right here would be the bundle of His, and the bundle branches right here, and then turns into the Purkinje fibers. Okay, now we've already mentioned that the SA node is most important. It is the fastest, so therefore that's what controls the contractile activity of the heart. Okay. Next, we're going to talk about the speed. So SA node, we mentioned, is already fast. AV node comparatively is slower, and then finally, Purkinje cells and the bundle cells are pretty fast as well. Okay, so just to make sure you guys all have it, SA, AV, bundle of His, bundle branches, Okay, now which direction do you think the electrical impulses travel? If you have to guess. Exactly, travels into different color. Blue. Here was the AV node. Now we mentioned that the AV node was slow. Okay, we'll get into the importance of this as we go on. But what happens is right where the AV node is situated, you have what's called fibrous tissue. What this does is this prevents the electrical impulses from going straight into the ventricle. Because what do you want to happen? You want the ventricles to fill up with blood, then pump. You don't want the impulses to just go everywhere and the heart just contracts without enough blood. Okay. So what happens is these fibrous tissue right here, it prevents the electrical impulses from just passing straight through. So the only way it can get to the ventricle is through here. Is the AV node. What it does is the AV node holds the charge, and then after a little while, it lets it go. And these cells, obviously, we mentioned these are fast, and therefore it conducts electricity fast in the ventricle squeeze. Okay. So that's the general direction of the electrical fibers. Now, briefly, we are going to get into the two different kinds of tissue. Okay. So, yes. So, we mentioned that before I go into it, what is the heart made out of? You know what the cells are called? Exactly, they're called myocardial cells, okay? And so these myocardial cells there's two basic different types. So let's put down here myocardium, okay, 1 and 2. So we mentioned that they were what? One functions as mainly contractile. So its main function is just to squeeze, okay? And it sucks at conducting electricity. So its main function is mechanical, all right? And so these are what? The atrial and now the other kind that we mentioned were the very specialized one, okay? Very specific to the heart. And these would be what? AV, or the SA would be first. AV, bundle, this, and bundle, branches, and Purkinje. Okay. Now, which one of these is the fastest? SA is correct. Fast and auto. Okay. Now, these are all automatic as well. So now, here's a question: if all those are automatic, why don't they all just, they have different levels of activity, so why don't they all conduct impulses at random times? Well, let me phrase the question again. Why is the SA node the main one? If these are all automatic, why is the SA node? You're right, someone does need to be in control, but that's not it. The main reason is because the SA node we mentioned is the fastest. Okay? So basically, whoever is generating this impulse first beats everyone else. If you think about it. Exactly, when it takes off. So that's why you have the SA node as the main one. It generates the impulse before everyone else. Therefore, it controls the contractile activity of the heart, basically. Technically, I guess if the SA node did get damaged, the AV node could take over. The AV node could damage, the bundle disc could take over. Obviously, your heart wouldn't beat as fast. Now, how many times do you think the SA node sends impulses per minute? It's around 100. Okay, it's around 100 impulses per minute. So now my question is, why, if it's 100 impulses, basically means 100 heartbeats, right, per minute. Now, why do you not have 100 heartbeats per minute? So what in your brain? You're correct. It's not exactly the brain, but it's I guess it's from the brain stem. So it's actually from the vagus nerve. Do you remember that? It's called the parasympathetic innervation. And we'll go over it later. But basically, you have the vagus nerve. Let's say, yeah, exactly. So let's say here's the brain stem, the spinal cord. Your vagus nerve comes in. And it's called parasympathetic innervation, and it brings this down to about 70 beats per minute. This is the normal heart rate that we all know about. And obviously, this is on a continuum. So when you're exercising, when your body cells need more energy and they need more oxygen, it beats faster. Therefore, you have a higher sympathetic innervation. And we'll go over that later, okay? It varies. Heartbeats varies for people. For some people, the normal is 60. Some people it's 70. Some it's higher. Obviously, when it gets too high or too low, then it's pathological, and they need to get that treated. Okay. So next, I'm just going to briefly talk about where the heart is situated and where these myocytes are located. Okay, we always mention the heart and the activity, but no, people have a hard time picturing where exactly these cells are. Okay, so the heart is actually situated in what's called pericardium. Okay, now pericardium is a double-layered sac. Okay. The best way I can show you in a picture form is if you can think, let's say you have a balloon, right? And you punch the balloon, you put your hand through the balloon without bursting it. So here's your balloon, okay? The opening of the balloon, and here is the fist, okay? And basically that's quote unquote your heart, okay? And so that's the pericardium. And so it has the outer layer of the pericardium, which is called the fibrous, and this is the visceral. Okay, now I am just briefly going to tell you where exactly these myocyte cells are located, okay? They're actually, in the picture of the heart, let's just span this picture over and briefly divide it into the different layers. So first you have your heart, okay? Here's the ventricle, here's where all the blood cells are. Okay, and then the immediate layer after that is that's the endocardial layer, okay? And then you have the myocardial layer, and then you have, immediately after the myocardial layer is the visceral layer of your pericardium, okay? So here's where the first visceral layer of the pericardium is in, okay? And in between the visceral and the myocardial, you have the it's called the pericardial cavity, okay? Now this cavity is actually filled with fluids, so your heart can beat smoothly to reduce friction. And sometimes if there's not enough drainage, the fluid can enlarge, impeding heartbeat, and thereby pathologically producing what's called a cardiac tamponade. Okay, and so you have your visceral layer, and then right after that, of course, is the outer parietal layer. Now we mentioned that this was the endocardial layer. Okay. And here you have the myocardial layer, and this is where your myocytes are, and that's where the electrical impulses get transported and they get conducted. Okay. Now, how exactly do these electrical impulses travel from one cell to the next? Here's one cell. Here's another cell. How would you imagine they travel? How does it? Close. Here, let's say that's an electrical impulse. How does it get through to the next one? They don't exactly shake hands. They have these gap junctions. Okay, and basically these gap junctions are quote-unquote holes in between the cells, actually with a transmembrane protein that helps bridge the gap, and the electrical impulses travel from one cell right on to the next cell. And we'll go into much more detail about how this happens next. Okay. Let's see. This next part, we're going to talk about how the cardiac cells maintain resting membrane potential. Okay, so talk about resting membrane potential. Okay, so heart cells need to maintain a certain level of electrical potential. Therefore, when it needs to conduct an impulse, it can change it and make the impulses go through. All right. So let's first take out. Okay, well, let's first talk about the what happens in one cardiac myocyte. Okay, now it's very important. This is a myocyte. It's not an AV node or SA node. It's a little different. I'll go into that in a little bit. Okay, how the heart maintains resting membrane protection. It's very simple. Okay, let's just imagine that this is a cell. All right, and if you had to guess, what would you guess maintains the resting membrane potential? Exactly, the sodium-potassium pump. Okay, and so here's a pump, it's transmembrane protein. What would you think it does?
What?
Sodium-potassium pump.
Like sodium, potassium.
Exactly. Now, what it does is it always pumps potassium into the cell and sodium out of the cell. Okay. So it's 3 sodiums out and 2 potassium in. Okay, and so now doing that, this would produce a net negative transmembrane potential, right? By doing that, so it's around, say, -5 millivolts. Okay, now the normal resting potential for a cell is around -70 millivolts. Now, how exactly do you think that resting potential gets there? Exactly, something else does happen. What happens is these cells have what you call sodium and potassium channels. Okay, normally the sodium channels are closed, so sodium can't go in. What it's creating is a concentration gradient where there is a higher amount of sodium outside and a higher amount of potassium inside. Okay, now if there's a higher amount of potassium inside, what they do is they just create these potassium channels whereby the potassium can just leak out. It's called a potassium leaky channel. And now when you have multiple channels like so, what happens, all the potassium leaks out, thereby getting -70 millivolt. So now you have to think of all these heart cells, right? They have this negative potential created by this potassium channel. Exactly. All right. Now, next we're going to talk about how an action potential is generated. It's simple enough to realize how the resting membrane potential. Now we're going to talk about how exactly this heart cell, cardiac myocyte, would generate a action potential. Go with a different color. We shall go with, okay. Alright, it's a green. Alright. Very, very simple. Alright. Action potential. So, on the side here, we are going to have a little graph. Okay. And we mentioned that the heart cell is around -70 millivolts. Actually, did I say -70? I meant -90. Okay, I apologize. It's not -70. It's -90 millivolts. Right. So here, let's say we have around -90. That's around -70. And let's just say it goes all the way to 0. Okay, we're just setting up a graph here. So this is time, and so here you have heart cell initially sitting and resting. Okay, it's called resting membrane potential. Now, what happens is the heart cell gets an outside kickstart, basically, right, from an impulse. And where would you think this impulse would come from? Exactly. But before, what would generate that impulse? We mentioned it first. Exactly, the SA node, okay? So now let's say that this cell is sitting right next to the SA node, okay? The SA node generates the impulse. Now the impulse travels and it hits this cell, okay? Once this cell gets hit, what happens is the membrane potential changes, okay? And now it's this change in membrane potential among the different cells, okay? One after the other, it's like a domino effect. That's how these cells transport quote-unquote, transport electrical energy, okay? That's how they generate these impulses and maintain it. So once this electrical impulse goes in, now we mentioned that it was negative. So what happens is this SA node generates a positive charge, a positive current, excuse me. And so this positive current hits this cell, okay? And now what it does is, exactly. What happens is they have the cells, the cardiomyocytes have what's known as a voltage-gated sodium channel. Okay. Now we mentioned that sodium was what? Had a higher concentration outside, right? Now the cell's pumping all this sodium outside. Okay. Now what happens is when this electricity hits it, the sodium channel recognizes it, and normally sodium channels are what they are in an it's what's known as a closed state, basically, where it's closed. But once this electricity hits it, it's going to go into an open state, okay? And these are the two lids, quote unquote, of the channel. And basically, all the sodium, which is collected outside, is going to start rushing in. Okay? Now, these channels, these transmembrane proteins are automated where after a certain mole they start closing, therefore it can't let further charges in. And so let's go to this graph. As soon as these sodium channels open, the sodium is going to start rushing in, and there's going to be a net positive charge on the inside, localized, okay? So this is going to go up, and now what happens is there's going to be a certain point where there's enough positive charge that it triggers this chain reaction of these channels opening. Okay, and it's going to conduct all the way to the next cell, the next cell to the next cell to the next cell, and then therefore generating and maintaining that electrical impulse. Okay, now that's called the threshold potential. It's right here at 70, and so when that happens, when it hits negative 70, it's going to go jump. It's going to shoot straight up, okay, and go all the way up to around 0, let's say. Now, once that happens, the sodium-potassium channels, they go from an open to closed. Now, they don't just close. What they do is they inactivate. So this bottom part right here, let me draw that again. So the bottom part right here closes off, the top part is starting to close, and so sodium can't go in anymore. Okay? And so once it hits that, these start closing. Now as soon as they close, there's another channel that opens up. Actually, there are two channels that open up. What would you guys guess? Okay, two channels, okay? They're called voltage-gated calcium channels and voltage-gated potassium channels, okay? And so what happens is, in order to bring the cell back to its resting membrane potential, these two channels open up. Now you have to realize that the cell also maintains a net. It tries to not have much calcium inside the cell, okay? It sequesters calcium in the sarcoplasmic reticulum in certain cells, or it usually pumps the calcium out. Okay, so there is a net negative loss of calcium in the cell. So there's a high calcium concentration outside the cell. So now once these calcium channels and the potassium channels open up, they both start. Okay, and so the calcium starts to seep in. As soon as the calcium starts to seep in, what happens? Calcium is positive, so what happens to the electric potential? Well, this is negative, so. Exactly, you're right. So I am sorry, that was incorrect. Let me correct myself here. Actually, you're right. There's no need to be confused. I was right. The calcium actually does go in. Now, instead of it becoming positive, what would happen? I told you the potassium channel is open too, right? What would happen to the potassium? Exactly. Potassium starts flowing outward. So what happens is it actually negates each other. It creates this sort of semi-plateau. Okay? Thereby maintaining that charge at that level. Okay? And so the calcium channels next close off, while the potassium channels remain open.
So that drops.
Then it drops. Okay? And it goes back to the resting membrane potential. Okay? And so that's how an action potential is generated in a cardiac myocyte. Yeah.
Excuse me, could you label the parts in the graph for me just so I can have a better sense of what happens where?
What do you want me to label?
So the resting part, the threshold, the super jump, the plateau, and then coming back down as individual parts, and what's responsible for.
Okay, well, here you have resting. Okay. And what happens is, remember we mentioned that the cell gets an outside positive impulse,
positive charge,
and now that's what makes it go up. Okay, so here's where the SA node hits it. Okay, and this is called the threshold. And once that happens, all the channels start going off in like a chain reaction, and it hits this point where the sodium channels close. This is the point where calcium and potassium open, and that's where calcium closes. Okay? You're welcome. And obviously the potassium continues to leak while sodium potassium pump works, and you have back to your resting membrane potential. Okay. Now that was what happens in a cardiac myocyte. All right. Now could you tell me why you see this effect? Why not just bring it back down? Why is there that plateau? Sort of, yeah. But why would you think that it would have that sort of plateau? What's the reason for the plateau? Okay. Well, the main reason the cardiac myocytes have that plateau is A, to prevent the cells from contracting again. Okay. So now the SA node is continuing to fire. All right, and let's just say the impulses are coming in rapidly. If these cells immediately go back down, then they would immediately fire again. They would contract. Okay. Now, if these cells keep contracting rapidly, the blood doesn't really get a chance to be pumped outside in one cohesive effort.
Oh, so that's the body's response to the
Exactly. Basically, that's the body trying to have enough blood in the ventricle, therefore squeezing, and in the atria. It's the same thing. It's just in the atria, it's not this long. It's a little short. The plateau phase is a little shorter. Okay. All right. So now we're going to go real quick into how the SA node works. All right. So we already mentioned the physiology behind the myocyte. Now let's talk about how the SA node works. All right. This is pretty much the same for the AV node as well, except obviously we mentioned that it doesn't go as fast, right?
Okay.
So let me draw the AV node. I'm sorry, the SA node. Here's the SA node the same. Okay. Now, the SA node doesn't generally get outside impulses. Okay. The only time it gets that is from the parasympathetic and sympathetic innervations, the outside innervations that control the heart. So, how do you think the SA node maintains its electric potential? Very good. Once again, the SA node exactly as the sodium potassium pump. So now, what goes in and what goes out?
Going out, potassium.
That's correct. So potassium, the body is trying to accumulate as much potassium inside the cell. Therefore, what's the reason for accumulating potassium? The end result being it wants to kick the potassium out in order to generate that current. Okay. Exactly. Exactly. So the way to remember it is the cell wishes to kick potassium outside the cell. It's okay, kick and potassium out. Okay. Actually, that's kind of confusing. Don't worry about it. This is the sodium. Out. And potassium goes in. Okay. Therefore, the SA node maintains a negative membrane potential. Now, what do you think the normal membrane potential is for the SA node? Actually, it's lower than the myocyte. It's well, I guess higher, not exactly lower. Yeah, the myocyte, if you know it actually. I'm sorry, the SA node actually maintains its membrane potential at around -60. Okay. Now, obviously, at this low level, it's kind of hard to at this level, it's kind of hard to generate that electrical impulse that the cardiac myocyte does. Okay, it uses a slightly different mechanism. Therefore, this is how the SA node maintains that automaticity. Okay, this is really important to pay attention. So, what's different in these cells? Okay, not only do they have potassium leaky channels, all right, but they also have sodium leaky channels. Okay. So, since the sodium, since SA node has the sodium leaky channels, it makes, it maintains it at -60 volts. Okay. So it's kind of hard for the cell to maintain the impulse like the regular myocyte would. Okay. Now, how would the regular myocyte go from this negative potential to a positive potential? Right, and then what are the molecules, the ions that travel into the cell to make it positive? Right, it's the voltage-gated sodium channels, right? Remember that? So the voltage-gated sodium channels open, and the sodium starts rushing into the cell. Exactly, and it goes up. But here, these cells already have a sodium channel. They're not voltage gated, they're just leaky channels. So it's constantly leaking out sodium. Okay? So it's kind of hard to have a voltage gated sodium channel. Okay? So now, instead of that, what the SA node actually has, it actually has a voltage gated calcium channel. Okay? That's kind of confusing, so I'll make it real simple. It's very easy. So, now, when this sodium potassium pump is working, it's going to maintain it at around, let's say, -60. Okay? The -60 millivolts. All right? So now, it also has these sodium leaky channels. Now, what would happen if the sodium continues to leak out? What do you think would happen here? Let's say it's -50, and there's like -45, right? What do you think? Does it go up or down? Exactly, it starts going up. So here it is at negative 60. Slowly it's going to start creeping up. Why? Because sodium is continuing to leak out. Correct? Even though the potassium channels are working, sodium potassium pumps are working, these channels are open. There's multiple channels. Okay, so there's multiple sodium channels, and sodium is continuing to leak out like so. Okay, so now what's going on is that there is a, I'm sorry, sodium's not leaking out. I'm sorry, sodium is leaking in, right? So the concentration gradient.
That's why
I'm sorry. Sodium is actually leaking inwards, right? Because the concentration outside is higher. Okay, my apologies. So the sodium is leaking in, so therefore there's this net positive charge that's automatically being generated. Okay, that's the key. These sodium channels are always open. That's how the SA node maintains automaticity, is because this is slowly creeping up, creeping up, creeping up. And once it hits around negative 45, this threshold potential, this is the threshold potential for the SA node, there's what you call voltage-gated calcium channels. Okay, well, voltage-gated calcium channels open. Now we mentioned that calcium is higher outside the cell, not inside the cell, right? So the calcium, calcium starts rushing in. You know, there's even more positive charge.
it becomes much higher.
It goes all the way up, right? And once again, say this is around 0. This is an approximate, it's not exact. And once it hits 0, the voltage-gated calcium channels start again inactivating. Okay, and then the net charge starts dropping again, and it comes back down to, because not only does the calcium channel start closing off after a certain period of time, we also have our friendly potassium channel. Okay, so that's how it works. In the SA node, in the cardiac.
What's happening in the potassium channel?
Well, what would you think would happen in the potassium channel? Where is exactly? Remember?
So it's kicking out the potassium.
It's kicking out the potassium. Okay, that's what I mentioned earlier. So all this potassium is starting to come out, and that's where you see this drop.
Okay.
Okay. So let me mark it here. So initially you have this negative 60, which is maintained by your sodium potassium pump. The sodium channels make it start creeping up. Exactly. Once it hits threshold, you have your calcium channels. Okay. And now at this point, the calcium channels automatically start closing. They're just programmed to close after a certain amount. At the same time, all right, potassium starts being kicked out. Okay, I hope you guys can see that. So it's very simple, and that's how the SA node maintains automaticity. Okay, now this continues. Now, over and over again, exactly. All right, and now once this charge, let's say, gets to the end of the cell, this is where it goes to the cardiac myocyte. Remember we mentioned that myocyte earlier?
Yeah.
And so the myocyte would get that external signal, the calcium would flow into the other myocyte.
Oh, yeah.
Right? And then the myocyte starts conducting the impulse. Correct? Or am I wrong? Next, we will talk about the atrial cell depolarization. Very, very simple. All right, so I'm just going to make a quick drawing, okay? So you have the atrial cell right here, okay? And we already saw how the SA node is generating that impulse. Okay, here's the SA node, bam, generates that impulse. And what is this impulse? Is it positive or negative?
Positive.
Positive, exactly. All the cells in the heart maintain sort of negative resting potential, right? Exactly. So once this positive hits, what do we have here? What kind of channels do we have here? Voltage-gated sodium channel. Exactly. Sodium starts leaking in. There is a net positive, all right? And then obviously you have, once it reaches that peak, you have your calcium channels that open up, and then the potassium channels at the same time, right? Potassium starts being leaked out, right? Before it maintains that plateau, right? And then the calcium channel closes, and then it comes back, all right? So if you want to look at a graph, it would look something like this. So here you would have that resting potential, and then boom, sodium comes in, sodium closes, potassium opens, starts being negative, but then calcium opens as well, so it goes to plateau. Since it's in the atrial cell, it's a little smaller, the plateau is a little smaller, and then gradually comes down. Once again, it gets another impulse from the SA node, boom. Okay, and it continues, so on and so forth. Okay, now briefly we're going to look at the different... This is the atrial cell.
Atrial cell. Okay.
Atrial myocyte cell. Remember we mentioned that there were two different kinds, specialized in the contractile. So this is the contractile cell, right? The myocyte in the atria. Okay. So this is basically the heart. This is basically a cell from here to here. Okay, we already talked about that, of course. Now I'm going to do a brief comparison of the different graphs, all right? And then we are going to talk about the parasympathetic and sympathetic innervations, and then we'll be done. All right, so first, let's look, sorry. Start with SA, then we shall look at ABV, look at an atrial cell, we'll look at Purkinje cell, and then we'll look finally in the ventricle. All right, so it's very simple. Remember, the SA node, how did the charges go? So you have that creeping up, all right? So slow, the sodium starts leaking in. Once the threshold potential hits, boom, you have the curve, and then the potassium opens. Calcium opens here, generates that spike, and then potassium opens, bring you down, potassium closes, and then once again, resting membrane potential slowly starts creeping up, boom, hits, down, and it continues so on and so forth. Now, the AV node is very similar, okay? Now, very similar to that. Okay, it holds, remember, it holds the charges and then it lets go.
Yeah.
It's very similar. It's holding, boom, it's holding. Okay, very similar. Now, what's different in the atrial cell is there is no this gradual creeping up. There's this constantly maintained resting membrane potential, but once that hits, all those sodium channels open, you have that immediate spike. You have this plateauing, comes back down, immediate spike, plateauing, comes back down, spike, comes back down. Notice it shouldn't be this long. The atrial cells don't have it, don't maintain a plateau for that long. And now, remember, what did we mention about the Purkinje cells? Were they slow or fast? Fast, right? So the Purkinje cell would have a very similar profile to the atrial cell, but it's also automatic. So it would have what, a similar profile to SA as well. So it would be some sort of like a hybrid. So we slowly creep up, and then we have this spike, plateau, come back down, creep up, spike, plateau, come back down, creep up, spike. Okay, something like that. Where you can still see that creeping up effect. Affection, we call it. And finally, you have your ventricle. And we already mentioned that. It's very simple. Spike, long plateau. Spike, long plateau. Spike. Okay. And that's basically it. So I'm just going to briefly go over the characteristics. So you have the creeping up here, which is maintained by the sodium leaky channels. Is that right? And a very slow rate of going up, maintained by what? The voltage-gated calcium channels, right? And you have the ventricle and the atrial, which are very fast, boom, boom, and the Purkinje, which is some sort of like a hybrid. All right, very simple. Now, very briefly, I'm going to talk about the parasympathetic and sympathetic innervation of the heart. Okay, so you have the autonomic nervous system regulation of the heart. All right, how does it do? How does it do? So we mentioned that there's two different kinds of autonomic. Excuse me. Exactly. Very good. Okay. Parasympathetic and sympathetic. All right. Now, what are the innervation of the sympathetic nervous system? What is the innervation of the sympathetic nervous system? Okay, so it's unlike the parasympathetic nervous system, or the parasympathetic innervation, which is basically from the vagus. This is not localized to one single cranial nerve. It's actually from it comes out of the sympathetic trunk and the cervical and thoracic nerves that come out of your spinal cord. Okay, and so you have that sympathetic innervation. All right. Now, what do the sympathetic neurons that innervate the heart? What do they? What is the neurotransmitter that they use to speed the heart up? Okay, it's norepinephrine. All right. Norepinephrine, sympathetic norepinephrine. All right. Now, it also uses epinephrine when it's circulating. So when you have hormones and such floating up to the heart, circulating in the bloodstream is epinephrine, and then the sympathetic innervation is norepinephrine. Okay. Now, what receptors do these the neurotransmitters? What do they bind to? You know? They're called beta-1 receptors. Okay. Beta-1 receptors. These are really important. So keep that in your mind. Let's go over here to the parasympathetic system. We already mentioned that it was what? It was vagal. It was this constant vagal tone. Of course, when it needs to lower it even further, there's an increased output from the vagus, therefore reducing it. Okay. So now the vagus, what it does, it mainly innervates the SA node and the AV node. It goes directly to the SA and the AV node. Okay. And what does this output? What kind of neurotransmitter?
Acetylcholine.
Exactly, acetylcholine. Okay. And what are the receptors that it binds to? Close. M2. It's called M2 muscarinic receptor. All right, and it's as simple as that. All right, so the vagus comes in, innervates, and in order to reduce the heartbeat, acetylcholine, sympathetic, it's norepinephrine, and it binds the beta-1 receptor. All right. Now we're going to just briefly talk about the effect that this does. How does this physiologically affect the SA node? Okay. First, we are going to look at sympathetic. All right. Then we'll talk about parasympathetic. Okay, once again, bring out the SA node. All right, so what you have is this transmembrane protein, okay? It's actually just 1, 2, 3, 4, 5, 6, 7. Okay, it's a 7 crossing transmembrane beta-1 receptor, okay? And, beta-1. Okay. And what you have is you have your norepinephrine or epi binds to this protein, and what this is, it's a G-coupled protein. Okay, so you have the G protein is a trimeric protein. It has alpha 1, beta, and gamma. Okay. Gamma. And once this binds, it's going to cause a conformational change, and the alpha and the beta gamma are going to separate. The alpha goes in, and it activates a protein called what? Adenylyl cyclase. Okay. Now, basically, the adenylyl cyclase converts ATP to cyclic AMP, and cyclic AMP goes ahead and binds to pKa. Okay, and basically pKa then phosphorylates. Okay, phosphorylates this transmembrane channel. Now, if you had to guess, what would this transmembrane channel be? What causes the heart to beat? What ion comes in? Remember? I mentioned that it was not sodium, but not potassium. Exactly, calcium. Right? So now this is how the heart, the sympathetic, modifies it. Once this phosphate binds this transmembrane channel, it opens. You have calcium come in. Remember what happens? Threshold potential goes up, and that's how it increases the heart rate, okay? So normally the heart rate, let's say, is something like this. What would happen with this is that when there's this innervation, we make it go a little faster, okay? So instead of waiting until here, we generate right there, faster. Okay, so here you only had 3, while it's red you have 1, 2, 3, 4. Okay, and even a 5th one after that. Now the parasympathetic is very similar to this, except what happens is instead of this being a B1 transmembrane protein, it is an M2. And what is this? ACH binds to it, and instead of this being alpha 1, it is actually alpha inhibitory. Okay, along with the beta gammas. And this is called a G inhibitory protein. Okay, G inhibitory. Earlier we mentioned sympathetic, that was a G activating protein. Okay, now what this does is this alpha goes ahead and blocks adenyl cyclase. So it effectively reduces the transmembrane channel being phosphatized. Okay, so phosphorylated, excuse me. And so it blocks that, therefore it blocks formation of cyclic AMP, blocks formation of pKa, thereby blocking the phosphorylation of the transmembrane protein.
Calcium doesn't come in.
Calcium is not able to come in because the channel is closed. All right, and therefore it actually slows down the heart. All right, now not only does it do that, but these two right here, the beta gamma, and they help open potassium. Well, what happens to potassium? As always, cell takes it out. Not always, excuse me. So the potassium channel pump, of course, brings the potassium in. So the potassium is being pumped out and thereby making it negative. Okay? So what would happen is that's how you have the normal heartbeat. And I'm going to use blue for parasympathetic tone. So it would be negative. It's not unable to reach that threshold potential, but slowly it does. Okay, and then it goes 5 slowly, so on and so forth. Okay, now is that clear? Okay. Do you guys have any questions? Okay. Well, if you have any questions, feel free to let me know.